This session focuses on understanding the following topic: Introduction to Microbial Genetics. If you've ever wondered how a simple bacterial infection can suddenly become untreatable, or how yogurt is made consistently around the world, the answers lie in microbial genetics. This area is fundamental for tackling major challenges like antibiotic resistance and is a frequent source of exam questions, particularly on mechanisms of genetic change. Let's begin. Microbial genetics, the big idea. The study of microbial genetics explores the compact but powerful genetic systems of microorganisms. Tiny genomes, big impact, reminds us that despite their small size, microbial genes drive processes from global nutrient cycles to disease. We'll answer how do microbes pass traits by looking at both reproduction and unique sharing methods. Why do antibiotics stop working is directly linked to genetic mutations and gene transfer that spread resistance. And finally, from curd starters to CRISPR, shows the practical spectrum from ancient food fermentation to the cutting-edge genetic tools derived from microbial defense systems. Meaning of microbial genetics. This field is defined as the study of heredity in microbes. Genes, traits, functions represents the central dogma where DNA sequences code for specific characteristics which then determine the microbe's role or behavior. It covers a wide range of organisms including bacteria, yeasts, molds, viruses. And it employs a combined lab plus bioinformatics toolkit, meaning we use both hands-on experimental techniques and computational analysis to map and understand genetic information. Why study microbial genetics? There are several key reasons. To understand microbial behavior, by decoding their genetics, we can predict how microbes will grow, respond to stress, or interact with their environment. To track resistance and virulence. This is crucial for public health, allowing scientists to follow the genes that make pathogens drug-resistant or more infectious. To improve fermentation outcomes. In industries like dairy and brewing, selecting the right microbial strains genetically ensures product consistency, flavor, and safety. And to design diagnostics and controls. Genetic knowledge enables us to create precise tests for pathogens and develop targeted strategies to inhibit harmful microbes. Genetic material in microorganisms. The primary genetic blueprint is DNA. However, RNA genomes in many viruses are an important exception, like in influenza or HIV. In bacteria, the genetic material is organized as a bacterial chromosome plus plasmids, where the main chromosome is circular and plasmids are small, circular DNA pieces that often carry extra traits like antibiotic resistance. In yeasts and molds, which are eukaryotes, we find linear chromosomes plus empty DNA, meaning linear chromosomes within a nucleus and separate mitochondrial DNA for energy production. The diagram shows a bacterial chromosome circular and smaller independent plasmids, highlighting the simpler organization of prokaryotic DNA. Genes, structure, and function. This slide includes core regions, a promoter where transcription begins, an ORF or open reading frame which contains the code for a protein, and a terminator which signals the end. The process is transcription translation, where DNA is copied to RNA, which is then read to build a protein. Gene expression is controlled at regulatory sites, operator, repressor. In prokaryotes, genes are often grouped into operons, clusters controlled as a single unit, while in eukaryotic genes contain introns, non-coding sequences that are spliced out of the RNA before translation. The table shows the structure of a gene and its RNA product. The first two rows show regulatory elements like the promoter and core, followed by the 5' UTR and the start of the protein coding region. The remaining values follow the same pattern and can be referred to in the table. This illustrates how a gene is transcribed into a pre-mRNA, which is then processed into a mature mRNA by removing introns and finally translated into a protein. Traits and inheritance. This slide connects the genetic code to the physical and functional traits we can see. Genotype-phenotype expression. The genetic sequence or genotype dictates the physical and functional traits of phenotype that we can see. Vertical transfer. Binary fission is the primary method where a parent cell splits into two genetically identical daughter cells. During this, correct chromosome or plasmid segregation is vital to ensure each new cell gets the necessary genetic material. Furthermore, environment modulates expression. meaning factors like temperature or nutrient availability can turn genes on or off, influencing the final phenotype without changing the DNA sequence. The diagram reinforces this, showing phenotype as the product of genotype interacting with the envir or environment. Genetic variation in microbes. Variation arises through several mechanisms. Mutations, point, indels, frameshift, are changes in the DNA sequence itself, which can alter or disable proteins. More impactful is horizontal gene transfer or HGT, where genes are shared between existing cells, not from parent to offspring. The three main HGT methods are transformation, where a cell takes up free DNA from the environment, transduction, where a virus phage accidentally carries bacterial DNA from one cell to another, and conjugation, where two cells connect via a pellis to transfer DNA directly. These processes are often aided by mobile elements, transposons, integrons, which are DNA sequences that can move within or between genomes. The diagram visually summarizes HGT. For transformation, a donor cell releases DNA, which is taken up by a recipient cell. For conjugation, a donor cell connects directly to a recipient cell by a bridge. For transduction, a phage-infected donor cell releases phages that carry bacterial DNA and inject it into a recipient cell. This slide presents a flow chart for cheese production and connects it to genetics. The process begins with raw milk, which undergoes standardization and sterilization. Key steps include add starter cultures, which are selected bacteria, add coagulant to form curds, cutting the curd, and steps like heating, discharging whey, molding, salting, and final maturation and storage. Different cheeses like Camembert, Romano, and Parmesan result from variations in this process. The second part, dairy microbiology applications, shows the genetic principles behind improving this process. We select robust starter strains for consistent fermentation. We use phage resistance genetics to protect cultures from viral attacks. EPS genes for texture refers to genes that produce exopolysaccharides, which improve the creaminess of yogurts and cheeses. Lactose pathway optimization involves selecting or engineering strains that efficiently convert lactose. And CRISPR typing for stability is a method to track and ensure the genetic consistency of microbial cultures over time. If this breakdown of microbial genetics principles was helpful, you might find the detailed exploration of other microbiology topics in future sessions equally informative.